Okay, this is podcast number 1 for Standard 3.2. This is the first of 3 podcasts, and in each of these podcasts we're going to be going over DNA replication, and then we will be talking about the molecule RNA, and then we're going to take a look at how RNA is involved with translating into a protein. So you would be able to identify the purpose of DNA replication, relate the concept of complementary base pairing to DNA replication, making it a very accurate process. We're going to compare the structures and functions of DNA and RNA, and we'll describe the general structure of RNA and the 3 types of RNA, which are messenger, transfer, and ribosomal RNA. We're going to take a look at transcription and how it's different from replication. And then we'll take a look at the process of translation and how translation is going to allow our cells to make new proteins. So these are some of the words that you're going to be seeing in here, like transcription, translation. We'll be talking about the different kinds of RNA, what protein synthesis really is. And basically we're going to be going through the entire process of from DNA to where it can replicate itself or not, to transcribing into RNA, to translating into a protein. So we're going to start with DNA replication. So DNA replication is a chemical process that is going to make an exact copy of DNA. So we're going to be duplicating our DNA during replication. What this allows for our cells to do is to preserve our gene sequences for many generations. Our DNA, remember, determines all of our traits and the way that our bodies function. So we want to make sure that we're passing on those basic body functions to our offspring, and we want to make sure that the gene sequences that we pass on are the same as our own. So DNA replication allows for that to happen. Now remember, DNA is always duplicated before the cell divides. So before mitosis or meiosis, or if we're talking about a bacteria cell, before binary fission, you're going to make a copy of your DNA every time. A cell cannot divide if it has not copied its DNA first, because if it did divide without copying its DNA, each of the new cells wouldn't have the right amount of DNA in them. Alright, so the process of DNA replication begins with a special enzyme called a helicase, and remember, we know it's an enzyme because it ends in ASE. Helicase is actually going to attach to the double helix of DNA and breaks the bonds that are holding the 2 strands together and basically unzips the DNA molecule. And what you'll end up getting are 2 replication forks. And what this means is you're going to have 2 places where the DNA can now copy itself over through this process. There is also another enzyme involved called DNA polymerase. And this one is going to bind to each of the new DNA strands that we just split apart. These two strands that were together, they're now split apart. The DNA polymerase is going to come in, and its job is to bring in the correct complementary DNA nucleotide on the strand so that we can make an exact copy. Okay, so we'll take a look at a couple of pictures of how this works. Okay. Now remember, we want to make sure that we're not making mistakes, so DNA polymerase also has the job of proofreading the DNA, and thereby it checks for mistakes, so it makes sure that the correct nucleotide has been brought in on the correct spot. And if it finds that one is not supposed to be there, it can either fix the problem or stop the whole replication process from occurring, and that would usually end up in the cell being destroyed in the end. So let's take a look at what this would look like here. So here we have an entire molecule of DNA. Okay, so helicase is going to first of all bind on here, and it opens up the DNA molecule, the original DNA molecule, and these are your 2 replication forks that you have. And then 2 DNA polymerase molecules will attach on. And what they're doing is they're taking free nucleotides that are available within the cell, and they're bringing in the correct one based on complementary base pairing. And this happens on both strands in opposite directions. So we're going in this direction down here for this growth. This DNA polymerase will keep going, and as it gets closer to the replication fork, the helicase will open it up some more. This one is growing in the other direction, so again, it's going to start growing it that way. So instead of just starting at the top of the DNA molecule, the beginning, and replicating it all the way down, we actually start in the middle and then replicate out to both ends. all the way out. Through this process, what you end up getting in DNA replication is two of the strands of DNA are going to complement each other. Okay, so remember our two original strands complement each other because of base pairing. A will only go with T, and G will only go with C. And what we're going to actually end up forming are two new DNA strands. But the two new strands do not go together. Each of the new strands that are going to be made are going to bind to one of the old strands. So again, this is just showing you one of the replication forks. So here we have an original old strand here. So this is an original piece. And over here, we're building a new strand coming in, complementing the original strand. Okay, same idea over here. We have an old strand that's coming down all the way here, and this new strand is attaching on to that one. So at the end of DNA replication, you end up with one old strand of DNA and one new strand of DNA. Okay? So again, here is just a really basic picture of this. So here would be a small piece of DNA composed of two original strands. It unzips because of the helicase, and the new bases get brought in with the DNA polymerase, and they go in the right places. And then you end up in the end with two strands of DNA. So you have two new molecules of DNA. Each molecule has one old strand and then one new strand. One old strand and one new strand. So that way you're not keeping the old ones together and you're not just putting the new ones together. This actually is called semi-conservative replication because we are keeping the original with the new ones, and they're less likely again to have mistakes when you do that. Alright, so this whole process of DNA replication is very accurate because of the complementary base pairing. So remember, that's Chargaff's rule. So A will bind to T, and that is because they have two hydrogen bonds that they can make between them. And G will bind to C, and that is because they have three hydrogen bonds between them. So A and C are not going to bind to each other because they don't want to make the same amount of bonds, so that's not a good pairing. So adenine will only go with thymine, and guanine will only go with cytosine, again because of the hydrogen bonds that they're able to make. And because of this, very few mistakes end up being made during this process. T doesn't want to come over here with G. Occasionally mistakes can be made, occasionally things will be put in the wrong place, and that's called a mutation. But for the most part, your cells are really good at making sure that everything goes where it's supposed to go, so you do not have a lot of mutations happening all the time. Okay? So throughout this whole process, very few mistakes get made. If they do get made, they're either fixed or the cell is destroyed before you can start making really bad mutations going along the way.